everyone. Hope everyone is doing very well in their lives. This is presentation on the family Reoviridae presented by Saurabh Shukla, Department of Veterinary Microbiology and the ANDUT College of Veterinary Science, Ayodhya. Let us talk about general characteristics of this family Reoviridae. The Reoviridae family is derived from respiratory enteric orphan viruses. Orphan means not associated with any of the disease. It's family which affects both respiratory and GIT. Talking about its genetic character, it is double-stranded RNA virus with the symmetry icosahedral and is non-capsulated. As you can see, two of the consisting virus of this family, blue tongue virus and the rotavirus. The causative agent of blue tongue virus is orbivirus and the rotavirus is the other one. Moving on to the first constituent of this family, the blue tongue virus. Let us talk about its characteristics. First of all, its structure, the protein capsid. The protein capsid of the blue tongue virus consists of outer and inner portion. It is made up of two layers. The outer portion is made up of VP2 and VP5 protein and the inner layer is made up of VP3 and VP7 protein. Talking about their functions, the outer capsid protein neutralizes immune response and the inner one is highly conserved and known as group specific protein. Talking about the susceptible host of the blue tongue virus. Blue tongue virus affects cattle, goat and sheep, but talking about the most susceptible host of this virus is sheep. The isolation of this virus can be done by blood collection when there is febrile condition and transferring into the lab with the anticoagulant, or inoculation in the embryonated egg, or via different cell cultures, for example BHK21 Vero cell line, C6 by 36 cell line. Next, moving on to the distribution or epidemiology. This virus affects wide variety of domestic animals through the insect vectors. The insect vector that impacts or that carry out the disease is culicoid family of insects. It has currently 24 serotypes. Serotypes is serologically distinguishable strain. Moving on to the clinical signs, elevated body temperature from 107 to 108 degree Fahrenheit. You will get mean peak viremia at the 6 to 7 days after the infection. At peak temperature increases and there is leukopenia. As you all know, whenever there is febrile condition, the WBC number comes down. That term is known as leukopenia and it occurs same in blue tongue as well. Elevated temperature leads to depression, thereby inappetence, hyperemia, congestion, swelled mucous membrane, ulceration of the lips and tongue. You can see clear picture of an animal, or simply saying goat, affected by blue tongue virus. Moving on to the pathogenesis. Since we all know most of the virus or all of the virus multiply or affects the lymph nodes, this virus is no different from them. The first site of multiplication is the regional lymph node. From lymph node, it spreads to other parts of the lymph nodes. It has strong affinity with the endothelium, periendothelial cells and the pericapillaries. Virus is also observed in reticuloendothelial cells of the lymph nodes. Talking about its diagnosis, how can one diagnose and tell whether this animal is affected from blue tongue virus? So as we all know, clinical signs and symptoms, lesions, history help us to diagnose or help us to get an idea of a disease. The next confirmatory test or other tests include antibody detection in the serum that can be done by agar gel immunodeficient test, neutralization test, immunoperoxidase test, enzyme assays like ELISA and RNA page. Talking about its prevention and control, since we all know prevention is better than cure, so we must focus on prevention part. Vaccination with pentavalent vaccine is must. Moreover, since we know this disease is spread by insect vector, so keeping the animal in dry places away from the breeding side of the vector is much important. Testing out the animal for blue tongue virus which has newly introduced in the flock. If we are bringing any new animal in the herd, that animal must be tested and must be quarantined or isolated from the herd such that the disease cannot spread to other healthy animals. Moving on to the next segment of this family, the next virus is rotavirus. Talking about its characteristics, the term rota means, it is a Greek word which means wheel. So this name rotavirus has been suggested because the outer capsid of this virus appears like a wheel. Therefore the term rota given to it. Its genetic material composed of 11 segment, that is double stranded RNA, and it can be separated by RNA page. RNA page is RNA gel electrophoresis method, you can see on the left side of the slide. Now, since we know it has 11 segments, so the segmented nature of the genome leads to emergence of new strain. Due to this, tremendous diversity have been observed. VP4, the capsid protein present in the outer capsid, is responsible for its virulence, hemagglutination, neutralization, and VP7, which is a glycoprotein viral neutralization protein, is present in this virus. The VP7 protein is also called G type, and the VP4 is also called P type of the protein. So far, 14G and 20P types have been found. Talking about its epidemiology or distribution, it is detected throughout the world. This virus is associated with acute gastroenteritis and diarrhea in mammals. It is transmitted via fecal-oral route and the respiratory route. This virus is relatively resistant to environmental factors. In temperate countries, it shows definite seasonal patterns. The peak of the disease is found in winters. Its epidemiology has mainly been done using electrophoretic migration pattern of double-stranded RNA. Analysis using electrophoresis provides evidence of its diversity. Clinical signs. The incubation period of this virus is 16 to 24 hours. Young animals may die as a result of dehydration, but most recover within 4 days. It infects epithelial cells of villi, causing atrophy, malabsorption. As a result, villi shortens and there is reduced absorption. Undigested lactose of the milk promotes bacterial growth, hence diarrhea. Moving on to its diagnosis, by detection of viral antigen in the feces. From feces, gold standard test by electron microscopy. Tests include ELISA, counter immunoelectrophoresis test, RT-PCR, co-agglutination test, and hybridization test. These were the diagnosis. Now this is simple diagrammatic representation of the ELISA and RT-PCR, how these methods work. Talking about last segment, that is prevention and control. The very much important part for the spread of disease, to control the spread of disease, its proper management, the proper management of the animal and the farms. Ingestion of the colostrum for short period of time gives 48 hours of continuous feeding. Ingestion of the colostrum for short period gives protection for 48 hours, while continuous feeding of low amount of colostrum gives longer protection to the animal. Talking about the recovery, by feeding water than milk at the onset of diarrhea. ORS is also given when there is dehydration to the animal. With this, I conclude. Thank you.